guest.
Am I audible?
Dr. Kirti Kumar, can you hear us?
Yes, sir, I can hear you now.
Okay, are you ready for the meeting?
Yes, sorry, I'm not able to connect it from my system, so I'm connecting it from my phone. So if there is any problem, please bear with me.
Okay.
So yes, you are audible. So shall we start?
Yes, sir.
Can we start the meeting, Dr. Kirti Kumar?
Yes.
Okay, Rajni.
So first of all, a very good afternoon to all of you, all the dignitaries, guests, faculties, faculty members, and my dear students. I, Dr. Rajni Solanki, welcome all of you for the today's webinar. Before beginning the webinar, let us virtually light the lamp of knowledge and wisdom to take the blessings from Goddess Saraswati. I request our technical team to play Saraswati Vandana.
सद्गमया तमसो मा ज्योतिर्गमया मृत्युर्मामृतं गमया ॐ शान्ति 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 या कुन्देन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा.
Thank you, technical team. Tuber and storage root crops such as potato, yams, cassava, sweet potato, sugar beets, etc. have high nutritional values and they offer high potential to overcome food security issues. The lack of information regarding molecular mechanisms that govern below ground storage organ development has limited the application of biotechnological strategies for improving storage crop yield. In this webinar, our resource person discussed about his research related to the role of RNAs and RBPs in potato tuber formation, storage root development in sweet potato, as well as different approaches employed for tuber yield in potato. Encouragement, enthusiasm, and excellence are one of the major aspects of many organizations with an objective of encouraging spirit of research and innovation. I, Dr. Rajni Solanki, feel privileged to welcome you and all on behalf of Sri Vaishnav Vidyapith Vishwavidyalaya Indore for today's webinar on employing the molecular mechanism of mobile RNAs and RNA binding proteins for yield improvement in tubers and storage root crops. Due to busy schedule of our Honorable Vice Chancellor, Sri Vaishnav Vidyapith Vishwavidyalaya, Dr. Upinder Sir, I would like to request Dr. K.N. Guru Prasad Sir, Dean Faculty of Agriculture, SVIG, SVIG Indore, for virtual welcome of our Chief Guest, Dr. Kirti Kumar R. Thank you, technical team. Now, I would like to invite Dr. K.N. Guru Prasad Sir. Now, I would like to invite Dr. K.N. Guru Prasad Sir, Dean Faculty of Agriculture, SVIAG, Sri Vaishnav Vidyapith Institute, Vishwavidyalaya, Indore, for welcome address. Please, sir.
Thank you, Dr. Rajni. Chief guest and speaker in today's webinar, Dr. Kirti Kumar Kondare from National Chemical Laboratory, Pune. Dr. Vinod Dhar, Head of the Agriculture Division, heads of the institutes, heads of the department, faculty members, and students. On behalf of Institute of Agriculture Science and also Sri Vaishnav Vidyapit Vishwavidyalaya, I welcome our guest speaker, Dr. Kirti Kumar Kondare, for this webinar. Dr. Kirti Kumar is a molecular biology scientist working on the improvement of crops, and he has a lot of experience of working outside India also. He did his PhD from England and has also worked as postdoctoral fellow in England before coming and joining ICER in Pune. Our agricultural department is about 4-year-old now, and so we are a new university with many new departments in their inception. The talk today will be focused on the molecular biology approaches to improve the crop yields, and that will be quite a technical approach involving the sophisticated and state-of-the-art methods and equipment. So our students will have an opportunity to get exposed to how researches are done in through the molecular biology approaches, especially on the tuber crops in which Dr. Kirti Kumar specializes. We are in the process of developing the laboratories in the agriculture science, and we look forward to have a molecular biology laboratory also to do these kinds of work in our research in the coming days. We are in the process of developing these laboratories, and I welcome Dr. Kirti Kumar once again, and thank you for this opportunity. Over to you, Dr. Rajni.
Thank you so much, sir. It's a matter of great honor and pride that we have with us a very proficient personality, Dr. Kirti Kumar R. Kondare, sir, Assistant Professor, CSIR and NCL, Pune, Maharashtra. Sir was graduated with BSc Biotechnology from Savitribai Phule University, Pune, and obtained his master's from University of East Anglia, Norwich, UK, and doctorate from Harper Adams University, Newport, UK. His area of specialization is plant physiology and biochemistry. Dr. K.R. Kondare sir has published several research papers in national and international journals. At present, he is working as an assistant professor at CSIR NCL, Pune, Maharashtra. So, without any further delay, I would like to invite a resource person for this webinar, Dr. Kirti Kumar R. Kondare sir, to share his knowledge on employing the molecular mechanism of mobile RNAs and RNA binding proteins for yield improvement in tuber and storage root crops. Over to you, sir.
Yeah. Good morning, everyone. Am I audible?
Yes, you are. Please.
Okay. Thank you so much for the kind introduction. It is really overwhelming. So, without taking any further time, I would like to start with my presentation. So, you have to share my screen, I guess. Okay. Is the first slide visible?
Yes, sir, it's visible.
Okay, fine. Just give me a minute. I have to see how I can hide the. How can I hide this? Okay. First of all, good morning to everyone. So today I'm going to talk about how we can utilize the mobile RNAs and RNA binding proteins for yield improvement in tuber and storage root crops. So myself, Kirti Kumar. So I'm working as a DST Inspire faculty in Plant Molecular Biology Group of Biochemical Sciences Division, CSIR, National Chemical Laboratory, Pune. Okay, so this is going to be a brief outline of my today's talk. So first, I'm going to talk about what are the mobile macromolecules, including mobile RNAs in plants, and then what are the functions of these mobile macromolecules. Then I'll talk about how these mobile macromolecules or mobile RNAs has led to the discovery of the RNA binding proteins, which acts as a cargo partners for these mobile RNAs. And then I'll move on to the what are the mobile RNAs identified in potato, and then respective RNA binding proteins, and what is their function in potato with respect to tuberization pathway. Then I will also talk about how these mobile RNAs and their RNA binding proteins can be utilized to increase the tuber yield in potato. And currently what I am working as a DST Inspire faculty in CSIR NCL Pune is to investigate the orthologs of these mobile RNAs and RNA binding proteins in storage root crops of important staple crops. So here specifically, I'm working on one of the storage root crops, sweet potato, and I'll update you about what is our current progress with respect to identifying the functions of these orthologs of mobile RNAs and RBPs in sweet potato. So as you all know, plants are sessile and they remain stagnant with respect to throughout their life cycle. And unlike animals and humans, so wherever there are the extreme environmental conditions, they can run away from the challenging environmental conditions, or they can take the necessary precautions. But plants are very poor, and then they have to remain stagnant there. They have to face these environmental conditions, so that they have to cope up with these changing environmental conditions by themselves. And then they have developed different mechanisms, and one of the important mechanism is the long distance signaling in plants. So this long distance signaling, what is it? So this long distance signaling helps plants to communicate from one tissue type to the other tissue type. So some signals produced in the leaf can be communicated to the what is happening in the root, and then similarly signals produced in the root can also communicate to the above ground part to have a better coordination to cope with these environmental conditions. So plant vasculature, or the vascular bundles, mostly comprised of xylem and phloem strands, and this xylem and phloem, they act as a conduit for the transport of these macromolecules. As you know, there's xylem, the main function of xylem is the transport of water and minerals, and phloem is transport of sugar molecules. So that is what we have studied in the textbook. But now with the progress in the research, now we know that xylem is also capable to transport specific phytohormones, even carbohydrates and amino acids, whereas phloem has been shown to be more advanced, and it is known as an information superhighway, and it can transport selective proteins, mRNAs, small RNAs, phytohormones, small peptides, and also some other compounds. So this is a review we published last year, where we have tried to identify what are the mobile macromolecules identified in different plant species, and see what is their physiological relevance at the whole plant level. So as you can see here, the first identification of the mobile RNA was done in 1995. And then in last 26 years, whatever the research has been done, we have tried to summarize different mobile macromolecules like mRNA molecules, proteins, small RNA, and RNA binding proteins. So what is the importance of having so many mobile macromolecules in plants, although from different plant species? So as you can see here, these mobile macromolecules, they try to regulate almost all the growth and development related processes in plants. As you can see here, some of the these mobile signals, they are involved in the regulation of the flower development, which is the important process with respect to seed development. So selective of these molecules are also known to regulate the shoot apical meristem maintenance and the development. Also, they are known to regulate phloem transport, and selective molecules, they can also control the leaf and the root development. They are also known to control seed development and the germination. In case of potato, it has been also shown that these mobile mRNAs and the proteins, they can also control the tuber formation in potato. Apart from these growth and development related processes, as you can see here, different biotic and abiotic stress responses are also controlled by these mobile macromolecules. So overall, they do have a very critical role to play to control the growth and development in different plant species. So the way these mobile RNAs or the mobile macromolecules were identified, so the discovery of this flowering stimulus, what we all know as a flow region. So this flow region signal was kind of identified in 1936, but the true identity of this signal remained elusive for last 75 years. And then, so what was known that some signal is produced in the leaf, and then it is transported through this stem, through the phloem to the shoot apical meristem, and then in the shoot apical meristem, they regulate the organ identity genes to regulate or induce the flower development process. So until 2005 to 2008, this signal was elusive, and no one knew like what is this signal. But now, now we know that these breakthrough reports, they have shown that indeed there is a protein known as flowering locusti, FT protein, as we all know, and it has been shown that both mRNA as well as the protein of this flowering locusti is mobile. So these mRNAs and the protein, they are synthesized in the leaves, and then they move through the phloem to the shoot apical meristem, and they induce the flowering process in the shoot apical meristem. So coming to the potato tuberization, so what is common between flowering and tuberization with respect to long distance signaling mechanism? So in 1981, a remarkable observation was made by Chailakyan et al., where what this group has found that when a flowering tobacco plant or the shoot of the flowering tobacco plant, when it was grafted onto the non-tuberizing potato stalk, it induced the potato tuber formation. So this classic example has demonstrated us that the signal for the flowering and the tuberization could be similar. So what is potato tuberization? So in the potato plant, the environmental cues are perceived in the leaves, and then signals are generated. These signals are then transported to the stem, to the phloem, to the below ground modified stem known as a stolon. So this tiny structure, and then under favorable conditions, this tiny structure passes through several stages of the developmental transitions to eventually form a mature tuber, what we call as a potato. So in last 2 decades, it has been shown that several factors can regulate this tuberization process in potato. So some of these factors are photoperiod. So based on the day length, like long day or short days, so depending on whether the day length is greater than 12 hours or less than 12 hours, they can influence the potato tuberization process. So it has been shown that short day induces the tuberization process, whereas long days, they inhibit the tuberization process. Apart from photoperiod, the phytohormones are also shown to play critical role to govern these stolon to tuber transition stages. And important phytohormones are auxin, cytokinin, GA, ABA. They regulate these developmental transitions in potato. Then there are mobile RNAs and RNA binding proteins. There is also a FT ortholog in potato known as STSP6A. So that protein is shown to be mobile from leaf to the below ground stolon, where it acts as an activator for the tuber formation process. Apart from these factors, there is also a transcription factor known as cycling dog factor 1, which is also considered as a important regulator of the plant maturity in potato. Apart from these factors, there is a small RNAs also, like microRNAs, microRNA 156 and 172. And then there are also phase siRNAs or phase short interfering RNAs. They are also known to play a crucial role to regulate the stages of the stolon to tuber transition. So in today's talk, I'm going to mostly talk about the potato mobile RNAs and then RNA binding proteins with respect to their role in potato tuber formation and our approaches to increase the tuber yield using these two mobile factors. So, the initially question was like, are there any mobile RNAs in potato? So it has been shown that the first mobile RNA identified from potato was BEL5, which is a BEL1-like transcription factor. And then later on, other mobile RNAs were also identified like BEL11 and 29, and then POTH1, which is a potato homeobox transcription factor. So the common fact about these mobile RNAs is that they all belong to this 3-amino acid loop extension superfamily of transcription factors. And it has been shown that in diverse plant species, this BEL and NOX, they form a heterodimer, and then this heterodimer controls the expression of the target genes. So BEL5 was identified as a mobile RNA in 2006. And later on, so in potato, there are 13 BELs identified. So out of these 13, BEL5, BEL11, and 29, they are the major or the abundant BELs in the potato, and they comprise almost like 2/3 transcripts of all the BEL families in potato. So then later on, BEL11 and 29 were also identified, which are the close orthologs of the BEL5. Then in 2012, a class 1 NOX genes mRNA like POTH1 was also identified as a mobile RNA. So in the next slide, I'm going to discuss about what are the experiments they have done to demonstrate that these RNAs are mobile, and then what is their role in the potato tuber formation process. So to demonstrate any mRNA is mobile, you have to find out a way to distinguish whatever the mRNA you are going to show as a mobile from the endogenous mRNA, because in this endogenous mRNA is also going to be present in the potato plant. So taking the benefit of the fact that when you are trying to overexpress any gene in plants, so you have a 35S promoter or any tissue specific promoter, then you have the coding sequence of the gene. Say in this case, it's a coding sequence of mobile RNA like BEL5 or POTH1, and then there is an Agrobacterium NOS terminator. So it has been shown that this NOS terminator sequence, it remains with the transcript when you have the respective overexpression lines. So taking the benefit of that, so people started doing the grafting experiments. So heterografts are prepared using the overexpression lines of BEL5 or POTH1, and then they were grafted onto the wild type plant's rootstock. And then, so in wild type rootstock, you should not have the tangent presence, but when it was grafted onto the overexpression lines of BEL5 and POTH1, these authors were able to detect these tangents of BEL5 and POTH1 in the root as well as the stolon tissues, suggesting that indeed these transgenic BEL5 and POTH1 mRNAs, they are able to move from the leaf to the below ground tissues like root and stolon. So, okay, they are mobile. Then do they have any function with respect to tuber formation? So they have generated the overexpression lines of BEL5 as well as the POTH1. As you can see here, so this BEL5 overexpression was achieved using a petiole, leaf petiole specific promoter known as galactose synthase. And this gas gas is a control. As you can see, compared to control as well as the wild type plant, the yield of the potato plant was almost increased by 2-fold in the BEL5 overexpression lines. Similarly, in POTH1 overexpression lines, you can see compared to wild type, the number of tubers as well as the tuber yield was enhanced by overexpressing the POTH1 in the potato. So apart from BEL5 and POTH1, as I said, BEL11 and 29 are also the close orthologs of this BEL5, and we wanted to study if their mRNAs are also mobile and if they have any relevance with respect to the potato tuber formation process. So similar to BEL5, we also found out that we have performed a heterografting experiment similar to BEL5 using BEL11 and 29 overexpression scions grafted onto the wild type rootstock, and we could indeed detect the presence of this BEL 11 and 29 transgenes. R is the root and the ST is the stolon. So three different plants, roots and stolon tissues were able to detect these transgenes, suggesting that indeed BEL 11 and 29 mRNAs are also mobile, similar to BEL 5. However, we found out that BEL 5 was shown as a activator of tuber formation because it enhanced the tuber yield. So we expected that since these are the close orthologs, they will also have a dated. When we generated the knockdown lines of these BEL 11 and 29, indeed the tuber yield was enhanced, as you can see in the images also. So number of tubers as well as the overall weight of the tubers was also increased, suggesting that unlike BEL 5, which was the activator of tuberization process, these 11 and 29 are the repressors, and they have a role to play in the tuber development process. So as I said, this BEL 5 and potash 1, they form a heterodimer similar to BEL-NOX interactions, and then they regulate the target genes through binding to the tandem TGAC elements in the promoter of the target genes. So in these two reports, they have identified the target genes of BEL transcription factor, and these are some of the important target genes regulated by BEL and potash heterodimer. So this is an example shown with respect to key phytohormones like cytokinin, gibberellic acid, and auxin. And what is known is that so this BEL 5 potash heterodimer, it requires a tandem TTGAC element in the promoters of the respective target gene. As you can see here, so in these different target genes of BEL 5 and potash 1, number of such TTGAC motifs are shown in this table, suggesting that indeed these targets are regulated by the BEL 5 and potash 1 heterodimer. So this is an important characteristic any gene should have if they are supposed to be a target of BEL 5. So so far we know that different mobile RNAs have been identified in potato, and they are transported from the leaf to the below ground modified stem stolon. But then considering that they are transported to the phloem, so phloem is supposed to be enriched with the RNAs, and these are the naked mRNA molecules. They will be prone for the degradation. So people started thinking about that. Is there any mechanism which will help them, or is there any transporter which will help to transport these naked mRNA molecules from this leaf to the stolon, and also provide the stability during their transport? So the question was like, are there any RNA binding proteins that will help in the transport of these mRNAs? So there was the first RNA binding protein identified in the Drosophila, and not from the plant system. And you can see, so that RNA binding protein was known as a stoffin, and stoffin is a RNA binding protein, and it is also referred as a polypyrimidine tag binding protein. And so there are two mRNAs known as Oscar and bicoid, and you can see that the Oscar mRNA is look. So this is a Drosophila egg, and you can see this Oscar mRNA is localized at the posterior pole of this egg, whereas this bicoid mRNA is localized at the anterior end of this egg. So the major function of this stoppin is to make sure that it transports this Oscar or bicoid mRNA to the respective target site, which will help to help with respect to their function in the organ patterning. So these experiments have also demonstrated that stoppin indeed binds to this Oscar as well as the bicoid mRNAs, and it transports it to the respective, like in case of bicoid, it will be transported to the anterior pole, whereas in case of the Oscar mRNA, it is transported to the posterior pole. So this stoppin plays an important role with respect to localizing these mRNAs to the target site. Apart from Drosophila, this PTB protein has also been identified from human, and it has been shown that they also bind to the mRNAs, and this function of this human PTB1, HNPTB1, is shown with respect to the alternative splice repressors. So majority of these RNA binding proteins or PTB proteins, they are known to be involved in the alternative splicing process in the eukaryotic systems. So taking the benefit of this Drosophila RBPs as well as the PTBs in human. Just a minute. So these classic examples in Drosophila and human has paved the way for the discovery of the plant RBPs. Before that, these plant RBPs were not identified. So considering the movement of these mobile macromolecules or mobile RNAs through the phloem, so the question comes like, if a particular mRNA is mobile, it should be detected in the xylem or phloem. Most of the cases, it has been shown that it should be present in the, or it's transported through the phloem. So different methods have been developed so that you can collect the xylem or the phloem sap and then identify the different proteins present in the respective phloem or xylem sap. So this is an example like how you want to collect the xylem or phloem sap. So it is recommended that in a given plant, like here examples are shown for cucumber, oilseed rape and the tomato plant. So you cut the stem of the plant like about 5 to 10 centimeters from the soil surface, and then the basal part of the stem is used for the xylem collection because that is more connected to the roots and then nutrients are coming from the xylem. Whereas the apical part or the shoot containing part is used for the collection of the phloem. And then there are different methods. You can purify the xylem as well as the phloem sap and then identify what are the proteins present in the xylem as well as the phloem sap. So what about the plant RBP? So these findings or these experiments like collection of the xylem and phloem sap has led to an interesting observation or an interesting finding in the pumpkin plant. So this is a picture of the pumpkin plant. And you might have seen that if you try to cut the stem of the pumpkin, like few mLs of liquid oozes out, and then it becomes an ideal system to isolate the xylem and phloem sap and then identify the proteins if we're interested in identifying the RNA binding proteins from the phloem. So CMRBP50 was the first RNA binding protein identified in pumpkin phloem sap, and it is also a polypyrimidine tag binding protein. So when these authors tried to identify this pumpkin phloem sap, of course they identified the CMRBP50, but to their surprise, what they observed, so this CMRBP50 was found as a huge complex which can contain 16 different proteins and the 6 different mRNAs. So this CMRBP50 was carrying these 6 mRNA molecules, and it was in complex with these 16 different proteins. So these are the list of the 6 mRNAs that were found to be bound by the CMRBP50. So at that time, the function of this mobile RNAs was not known, but now we know that at least the shoot meristemless STMP and GA sensitive phloem GAIP function is known with respect to the flowering as well as the shoot apical meristem development. So this mobile RNAs as well as the CMRBP50, it is synthesized in the leaves. It forms a ribonucleoprotein complex, and then this complex is transported from the leaf petiole through the stem as a RNP complex to the shoot apical meristem, where the target RNAs will be translated and they will do their respective functions. So based on these findings, so Cho et al. In 2015 wanted to understand what are the PTB proteins in potato. So this is a special structure of the human PTB1, and with respect to PTB structure, so there are 4 RNA recognition motifs known as RRM 1, 2, 3, and 4. And the specialty about this RRM 3 and 4 is it is the motif which is involved in the interaction with the target RNAs. So these 2 motifs, RRM 3 and RRM 4, they are very crucial with respect to their binding to the target mRNAs. So in potato, 6 PTBs were identified, PTB1, PTB6, and then other PTBs, out of which this PTB1 and PTB6 were similar to CMRBP50, which was identified in pumpkin, because they had this RRM4 and RRM3, whereas these other 4 PTBs, they lacked this RRM4. So probably based on that, they could not be really the 2 RNA binding proteins with respect to our mobile RNAs. and they have a major function mostly in the alternative splicing events. So in this Choita's paper, they have also shown that this PTB1 and PTB6, they indeed binds to the mRNAs of this BEL5 and then potassium mRNA, and then they helps to form a ribonucleoprotein complex in the leaf, and then transport it to the stolon to induce the potato tuberization process. So it has been also shown that, so one point I forgot to tell you in the last slide. So if you see this binding of the CMRBP50 to the target RNA, you can see there is a structure similar to like U, C, CU. So there is a cytosine uracil motifs, and these RBP proteins, they bind to the target RNA through the cytosine uracil or CU motifs in the target RNAs. And mostly these target RNAs, these CU motifs could be present in the coding sequence. It could also be present in the 5' as well as the 3' UTRs. And then in potato, it has been shown that the CU motifs present in the both 5' and 3' UTRs are very efficient one for the transport of this BEL5 or potassium mRNA to the target site. And if you try to delete these motifs, these mRNAs, they don't move from the leaf to the stolon. So you can imagine the importance of these 5' and 3' UTR. Again, the experiments have been conducted which shown that the 3' UTR is more efficient for the transport of these mRNAs compared to 5' UTR. So to understand further, what is the function of these PTB proteins in potato? So again, the same group, Cho et al., they have generated the overexpression as well as the knockdown lines of this PTB1 and PTB6 and evaluated their tubercle phenotypes. As you can see here, compared to wild type or the vector control, in the individual overexpression lines of PTB1 as well as PTB6, the tuber yield was enhanced by twofold. And this was accompanied with the, you can see, compared to wild type, the levels of BEL5 was significantly enhanced or up to twofold in the PTB1 and PTB6 overexpression lines. So PTB1 or PTB6 is overexpressed, which leads to the increase in the levels of the BEL5 transcripts in the stolon, thereby inducing the tuber yield. Similarly, so when the antisense or the RNA lines of this PTB1 and PTB6 were generated, you can see the tuber yield was significantly reduced, and this was also accompanied with the reduced expression of the BEL5 in the stolon. So indeed, PTB1 and PTB6, they act as a positive regulator for the tuber development, but their effect is indirect. It is mostly through the enhancing or decreasing the levels of BEL5 in the stolon, considering they are the cargo partners of the mobile RNAs. So based on this, we have proposed a model for the mobile RNAs and proteins in potato tuber formation. So I'm also going to describe about one of the protein, which is FT ortholog STSP6A, its role in the potato tuberization apart from this mobile RNAs and RNA binding proteins. So in this pathway, so these mobile RNAs like potash1, BEL5, BEL11 and 29, they are synthesized in the leaves. At the same time, these PTB proteins are also synthesized in the leaves. They form a ribonucleoprotein complex, and then this ribonucleoprotein complex moves through the phloem to the below-ground modified stem stolon. At the same time, part of the BEL5 and potassium mRNA, they are also translated in the leaf, and the BEL5 potassium heterodimer, it acts as a activator for this FT ortholog STSP6A, and as a result, this expression of this STSP6A is increased in the leaves, and it has been shown that the protein of this STSP6A is mobile, similar to BEL5 from leaf to the stolon. And then when all these mobile signals, they reach the stolon, they try to induce the tuberization process, and you get tubers formed from the potato plant. So when potato plant is under long day conditions, which is a non-tuber formation condition, so this is the elongating stolon. And what happens when these mobile RNAs, RNA binding proteins and mobile protein STSP6A reaches to the stolon? So as I said that BEL11 and 29, unlike BEL5, they act as a repressor. So when the stolon is not forming a tuber, that time this complex is prominent, and it helps to keep the stolon growing, and tuber formation process is not initiated. And the way it functions is this BEL11 and 29, they try to increase the bioactive GA levels in the stolon. So when the GA levels are high, stolon keeps on elongating and keeps on growing in the soil. And it does so by increasing the expression of the GA biosynthesis genes like GA3-OX and GA20-OX, and by downregulating the expression of the GA catabolism gene GA2-OX1. So this is under non-tuber inducing conditions. What happens under tuber inducing short day conditions? So you have two parallel pathways functional here. So first of all, the mobile RNAs like BEL5 and POTACH1, they are translated, they form the respective proteins or transcription factors. The STS6A protein is also there. So this BEL5 and POTACH1 forms a heterodimer, and then they regulate or they induce the expression of this GA catabolism gene, or they repress the expression of this GA biosynthesis genes. At the same time, this FT protein is known to induce the expression of this GA catabolism gene GA2-OX1. So these two parallel pathways help to reduce the bioactive GA levels in the stolon, and now stolon stops elongating and it starts to swell at the subapical region, which leads to the formation of the tuber. So these are the changes that happens in the stolon under non-tuber inducing condition of long day and under tuber inducing conditions of the short day. So based on these findings, what we try to do, we have taken a gene stacking approach using mobile RNA BEL5 and PTBs, PTB1 and PTB6, and we have generated 3 different chimeric constructs. So you can see in chimeric construct 1, so with respect to each gene like PTB1, PTB6 as well as BEL5, it is with respect to the plant-based terminators, and the coding sequence is driven by the respective gene's native promoter. And that is the case for all 3 genes. So in case of the CC1, chimeric construct 1, you have BEL5 cassette as well as the PTB6 cassette. In case of CC2, you have PTB1 and the BEL5 cassette, whereas in CC3 triple construct, you have all 3 genes. So here the idea is, so if you see, because they are expressed on the native promoter, so you expect already the plant is having 1 copy of the gene, and you are putting extra copy of the another gene, means same gene, but you are increasing the expression of that particular gene by 2-fold. So we expected that by having this gene stacking approach, can we increase the potato tuber yield, considering the functions of these mobile RNAs and the PTBs. So we generated the transgenic lines of these CC constructs, so double constructs and triple constructs. As you can see, the morphologically the plant looks similar to wild type plant. We also quantified the shoot biomass and the root biomass of these plants, and there is not significant difference with respect to, or I would say there is no common trend associated with respect to shoot and root biomass of these transgenic lines compared to the wild type. We also looked into the tuber bearing capacity of these transgenic lines. So tuber bearing capacity is one of the important traits that governs the potato yield, and it is nothing but a measurement that considers the how many number of stolons and tubers are produced by a particular potato plant. So more number of stolons and tubers, more likely the yield is going to be. So what we could see that in the triple construct, the overall tuber bearing capacity was significantly enhanced compared to the wild type, suggesting that this our approach could lead to the enhancement in the tuber yield. Then we conducted the greenhouse experiments using these transgenic plants, and as expected, we could see an increase in the tuber yield in all 3 types of constructs compared to the wild type. So on the left hand side, y-axis, you see relative tuber yield per plant, and in right hand side, y-axis is relative BEL5 levels. So we wanted to correlate that by expressing these mobile RNAs and the RNA binding proteins together, whether it has led to the significant transport of this BEL5 to the stolon. So indeed, we could see that all the transgenic lines, the level of BEL5 was increased by 2-fold, and the tuber yield increase was significant. It was in most of the cases 7 to 10-fold compared to wild type. To take these findings further, in collaboration with Central Potato Research Institute in Shimla, CPRI, we have conducted a field trial on these transgenic lines. So one more point I forgot was, so this kind of approach we have demonstrated in 2 different potato cultivars. So here I'm showing the data from a day neutral potato cultivar Desiree. So we could grow them in the field also, and indeed in the field trial also. So this is the first field trial data. We could see compared to the transgenic control, the yield of the these potato plants was increased up to 8-fold. and the triple construct was more significant compared to your double constructs. So indeed, the gene stacking has increased the potato yield by several folds. So this is about the potato, what we have done so far, what is known in potato with respect to mobile RNAs and RNA binding proteins. So now, as a DST Inspire faculty, I'm working on a storage root crop sweet potato and trying to understand, because considering the below ground tuber development in potato and also the storage root development in several storage root crops like sweet potato, cassava, carrot, radish, and sugar beet. So we expect that the mechanism could be similar, but although these storage root crops are nutritionally rich and they have diverse industrial applications, they have been research neglected for many years, and major reason was the non-availability of their genome sequences. However, in the recent years, there has been significant progress made, and some of the, not all the storage root crops genome is sequenced, but some of them has been sequenced, and now it opens up or it gives us a scope to understand the functions of genes that could be involved in the storage root development. So as I said, these storage root crops are nutritionally rich and they act as a staple food in many developing countries. Many of them, they have medicinal properties and they also have industrial importance. And this point I have already explained. The genomes of few crops are recently sequenced. And however, the storage root mechanism is as such not known at all. So at least, I mean, potato, which is a tuber crop, it is, I would say, well established, but with respect to any of the storage root crops, the mechanism of storage root formation is not known. So why we are using sweet potato as a model system for storage root development? Of course, the genome of sweet potato is not known. So the genome of Ipomea trifida, which is a diploid and is a close ancestor of the sweet potato, was sequenced in 2018. And actually the genome of Ipomea batata is 1.6 GB. And another point, why sweet potato? Because the agrobacterium mediated plant transformation system is known for sweet potato, although the efficiency is very less compared to other plant systems. So what are the different types of roots in sweet potato? So unlike potato, where the photoperiod plays a very critical role for the tuber formation, in sweet potato, which is a storage root crop, it's mostly the process is dependent on the age of the plant. So if the life cycle of the sweet potato plant is like say 3 to 4 months, so in the first month it produces only the fibrous roots, which are the adventitious roots, and then in the second month it starts to produce the storage roots. And at the time of harvest, they will also have a mature storage roots. And in these mature plants, generally you find 3 types of root systems. So fibrous roots are the adventitious roots. Then you also have 2 other types of roots, pencil roots and the storage roots. So these pencil roots, they remain like pencil-like structure throughout their life cycle of the sweet potato plant, and they have very high levels of lignin. Whereas the storage root crop, they have very low level of lignin and they have high levels of the starch. That makes them as a ideal staple food crop. So apart from this, so our previous studies have shown that indeed the orthologs of this BEL5, POTH1 and STPTB1 and PTB6 are also present. And apart from that, the FT orthologs are also reported in these storage root crops. So we wanted to understand like whether there is also a mechanism related to mobile RNAs and RNA binding proteins in sweet potato. So what we have observed that, of course, we have tried to study all these 5 storage root crops and see what information we have from these storage root crops. So we identified the orthologs of this PTB1 and PTB6 from 5 storage root crops. And when we did the multiple sequence alignment, what we could see that, as I mentioned earlier, that RRM3 and RRM4 of the PTB protein is very crucial with respect to their interaction with the target mRNA. So these 4 RRMs are highlighted by the red underline. You can see, so this is the first RRM, then this is the second one, then this is the third, and this is the 4th RRM. And you could see that so each RRM is known to have 2 RNP motifs highlighted in the yellow color. So these RNP motifs are the actual motifs which would bind with the target mRNAs. And what we could see that compared to potato PTB1 and PTB6, all the orthologs from these 5 storage root crops, although they are from different families, they do have conservation in the PTB orthologs, and there is also a conservation in the RRM structures as well as the RNP motifs, suggesting that indeed they could be doing a similar function in the storage root crops. Then considering that these PTB orthologs, they interact with the target mRNAs through the cytocellular cell motifs in the 3' UTR. So we try to look for this number of CU motifs in these orthologs of the storage root crops. And indeed we could see that you can see like number of such motifs were really high, suggesting that these orthologs identified from the 5 different storage root crops, they could be functioning as a mobile RNA similar to BEL5. As you can see, so there are 2 control bells here, BEL 14 and BEL 22, which are shown as non-mobile. They have very low number of such CU motifs, and that is the reason why they are non-mobile. Also, another point what we know in potato is that when you do the expression analysis of this BEL5, POTATE1, as well as this PTB1 and PTB6 in leaf versus root tissues, we could see a differential accumulation of these transcripts in the 2 tissues. And similarly, we checked the expression of these orthologs in 5 different storage root crops. And indeed, we could see a similar expression pattern, not similar, but I would say a differential expression pattern between leaf and the root tissues, suggesting that indeed this mechanism will be functional in these storage root crops. Then considering the fact that this BEL5 and POTATE1 heterodimer, they interact or they regulate the target gene expression through this tandem TGAC motifs in the promoters of the target genes. So we tried to search for the TGAC motifs in the sweet potato orthologs. And indeed we could see that, so these are the number of TGAC motifs in the promoters of the potato target genes. And these are the orthologs of these genes. We could see that such tandem motifs are also present in the sweet potato orthologous genes. So these results suggest that indeed there is a conservation in the target gene regulatory network that governs the hormonal metabolism related genes. So based on these findings, it really makes sense to study what is actually the functions of these orthologs and in sweet potato. So Aruna, who is a PhD student in my lab, she has identified 4 PTB-like genes from sweet potato. Along with that, she has also identified 6 BEL5-like orthologs and 4 POTATE1-like orthologs. And currently, she is taking different approaches to understand the functions of these orthologs in sweet potato. So tissue-specific expression of these orthologs. So mostly, we'll study the expression of these orthologs in leaf as well as the different root structures or root types like fibrous roots and the developmental stages of the storage roots in sweet potato, to demonstrate really these orthologs are really RNA binding proteins or not. So she's planning to do the EMSA, RNA EMSA, to test whether these orthologs of the PTB1 and PTB6, they indeed bind to the orthologs of this BEL5 and POTATE1. And we are also taking overexpression as well as knockdown and knockout approaches for the functional characterization of these orthologs. So this will help us to understand whether these orthologs have any function with respect to storage root development, and also it will also help us to understand the molecular mechanism regulated by them if really they have a function in the storage root development. So, so this is a general updated tuberization pathway with respect to mobile signals. In today's talk, I have mostly talked about mobile RNAs like BEL5, BEL11, 29, and POTATE1. But apart from that, this FT ortholog STSP6A as a protein has been also shown to be a mobile. And 2 microRNAs like microRNA 156 and 172, they're also shown to be phloem mobile. They are also synthesized in the leaf and transported to the stolon to regulate the tuberization pathway. So with respect to microRNA 172, it has been shown that it acts as a activator of tuberization process by inducing the expression of BEL5. You can see here BEL5 is inducing the, microRNA 172 is inducing the expression of BEL5. And another point with respect to microRNA 156 is that, so we have shown that if you overexpress this microRNA 156 in potato, you get to see this unusual phenotype of aerial tuber formation. So it has a scope to utilize this phenotype in the field condition. So we're trying to work on this. So to summarize my today's talk, we have shown that we can increase the potato yield by 2 to 3-fold in Bell 11 and 29 lockdown lines. Gene stacking approach has led to the enhanced potato tuber yield in the field conditions. And if we can identify the orthologs of these mobile RNAs and the RNA binding proteins in sweet potato, if they are having a function in the storage root development, then we can employ a cisgenic approach coupled with this multi-gene technology, and sweet potato yield also can be enhanced. So with this, I come to the acknowledgement. So I would like to thank Professor Anjan Banerjee for his constant support and guidance during my postdoc tenure. And also would like to thank all the past as well as the present members of the Molecular Plant Biology Lab at IISER Pune for their continuous support as well as the critical comments during the work. And at CSRNCL Pune, first I would like to thank Director CSRNCL Pune for hosting me as a DST Inspire faculty, and the current plant molecular biology members for their continuous support in my work as well as their continuous guidance. And of course, research cannot be completed without the funding. So during my postdoctoral tenure, I had 3 years of RA-ship from the Department of Biotechnology and 2 years of postdoctoral fellowship from IISER Pune. So thanks to them. And at CSRNCL Pune, I have a DST Inspire fellowship as well as the grant from the DST, and CSRNCL Pune for providing me support for my research. Thanks. So now I would like to take the questions if there are any.
Thank you, sir. Thank you, Dr. Kirti Kumar, sir, for such a valuable and fruitful session. Now the panel is open for discussion. If audience has some query, they may ask. Audience, if they have some queries, they may ask. Okay. Now I would like to invite our coordinator, SUIAG, SVVV, Professor Vinod Dhar, sir, for vote of thanks.
Thank you, Dr. Baba Sahib. Dr. Kirti Kumar, our chief guest in today's webinar, Dr. K.N. Guru Prasad, Dean, Faculty of Agriculture, Shri Vaishnav Vidyapith Vishwavidyalaya, directors, head of institutions, coordinators, head of departments of the constituent institutions of Shri Vaishnav Vidyapith Vishwavidyalaya, and dear students. Before I formally place my word of thanks, I would like to share with you all that Sri Vaishnav Institute of Agriculture is making all-out efforts to be in forefront of imparting quality teaching and gainful learning and employment in agro industry, ready and other agro-based industry. So the students are ready to serve the humanity. Students are being given exposure to hands-on training, rural attachments in their 7th and 8th semester, so that they pass out, so that when they pass out, they are ready to take up their own enterprise also. Well, I thank Dr. Kirti Kumar for his enlightening and thought-provoking lecture, quite academic in nature, and on role of molecular mechanism of mobile RNAs and RNA binding proteins in the yield of tubers and other storage root crops. I express my sincere thanks to Dean Faculty of Agriculture, directors, head of institutions, coordinators, head of departments of the constituent institutions, and particularly to Dr. Kirti Kumar R. Kondhare for sparing the time and giving us an opportunity to share what we are conducting at Institute. It's always our effort to invite eminent scientists to the forum for giving lectures and guidance, which will in fact serve as an inspiration to them. As the today's lecture has, I think the students must have felt the curiosity to go into the researches, and which is very vital. We must encourage our students to go in for the researches and ultimately in the development. So thank you very much, Dr. Kirti Kumar. Once again, I thank you for sparing the time and giving us such a valuable insight into the molecular biology. Thank you very much.
Thank you, sir. Now I request our technical team to play national anthem. So I request all, please rise.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you. With this, we have come to the end of the webinar.
Well, thank you, Dr. Baba Sahib Walunchkar and Dr. Rajni for conducting this program. Thank you very much.
Thank you, sir.
Thank you, Dr. Kirti Kumar. Thank you very much.
I would like to thank you everyone, for your support and giving me opportunity to present my work, because this is my first invited talk. Actually, I really feel happy.
No, we have just been enlightened by your lecture. It is really thought-provoking and has arisen curiosity in our minds and we will I definitely feel that students must have been very curiously listening to this lecture of yours. Thank you very much.
Yeah, I mean, so I feel that, so mostly our research is basic molecular biology, but then now we are seeing, considering the functions of these mobile signals, we do really have a chance to employ them for the agricultural productivity. So that is the emerging field now we feel. And potato, we have shown it. So maybe in future, so like I would share one more example, like with respect to rice and wheat. Also, there is one mobile RNA binding protein known as RBPP in rice, where it has been shown that it induces the yield of the cereals.
There could also be the possibility of these in turnip as well as in knoll cole.
Yes, that's what. So I'm taking sweet potato as a model here, but yeah, of course, down the line we also wanted to explore other storage root crops. So turnip is also coming in the
turnip as well as knoll cole rabi.
Yeah.
Okay.
So even ashwagandha is also considered as a storage root crop.
Yes.
So there was one question from the student. Just wanted to answer that. What could be the difference between DNA and RNA binding proteins? So at least for the RNA binding proteins, so I mentioned that this RRM3 and RRM4 is the important one, which will actually interact with this cytosine uracil motifs in the 3' and 5' UTRs of the target mRNAs. So probably that kind of motifs are not there in the DNA binding proteins. And then DNA binding proteins can be like transcription factors. They have different motifs, and because so these RNA binding proteins, they bind to the RNA. So you expect that RNA sequence will be there, like the CU motifs. But in case of DNA, these are the cis regulatory motifs, and mostly the transcription factors are the one which binds to this DNA structures.
All right.
Yeah. Thank you, sir.
Yeah.
Now I would like to thanks our chief speaker, Dr. Kirti Kumar sir, Vice Chancellor, Dr. Rupender Dhar sir, Dean Faculty of Agriculture, Dr. K. N. Prasad sir, Professor and Head and Coordinator of Department SVIAG, Professor Vinod Dhar sir, faculty members and students for being with us. Have a very good day. Thank you all of you.
Okay,
Thank you. Have a nice day. Yeah.
Alok sir.